Hello everyone, and thank you so much for joining me for today's lecture on the principles of pharmaceutical analytical methods and validation. So we're going to look into ICHQ2 R1, revision 1, and ICHQ6A, and obviously we are going to go over a few analytical methods that we use in order to prepare our regulatory documents. These analytical methods are useful, the knowledge of course, for writing module 2 for the quality overall summary document, and of course for preparation of the module 3 for submission. It can be an IND, it can be a new drug application, NDA, it can be a marketing application authorization for Europe, EMA, it may be for a CTA or clinical trial application for Health Canada, or it can be for new drug submission for Health Canada as well. So we are going to look into the analytical methods today. We are going to have a better understanding of what are those methods. So what are the learning objectives today? We are going to define the role of analytical procedures in the chemistry, manufacturing and control development, or CMC. Then we are going to try to understand the product-driven variability in required analytical procedures, distinguishing between universal versus product-specific tests. We're going to explain the importance of characterization across all stages of product development. We're going to understand the differences between analytical method validation characteristics. We're going to identify FDA guidances and ICH guidelines relevant to analytical procedures and method validation. And we're going to gain a basic understanding of the purposes, equipment, and operation of several widely used analytical procedures like HPLC, GC, and NMR. Okay, so let's have an overview of the analytical methods. So the overall purpose of analytical methods is to identify and to quantify. And of course, we will focus on methods for identification and separation. What is that exactly? Because when we are dealing with pharmaceutical products, we are making a product. That's a substance first. And of course, we use the substance, we add excipients, and we are going to have the final product. But in order to, prove that we are making the substance that we are talking about, that we are claiming, saying that this substance is going to have some, I would say, use in the healthcare sector, we will need to identify that. And once we identify that, we also need to use several methods in order to do the separation. Why? That's because in any chemical reaction, we are not just going to have exactly the substance that we are looking for. We're looking into having a substance, but in addition to a substance, we will also have some other products that are going to be made based on the chemical reaction. And of course, we call those impurities in this case. It means that now, after chemical reactions, we are going to have our substance plus impurities. First, we will need to identify our substance, and then we will need to purify that, to remove that substance from that mixture of the substance plus impurities. This is why we talk about analytical methods. So when we look into ICHQ2 R1, we can see the definition of analytical procedure, which is a method used to test a defined characteristic of drug substance or drug product against established acceptance criteria for that characteristic. So the four most common types of analytical procedures are the following. One is the identification tests. The second is the quantitative tests for impurity. The third one is the limit tests for the control of impurities. And the fourth one is quantitative tests of the active moiety in samples of drug substance or drug product or other selected components in the drug product. So we would like to quantify, we would like to know what are the substances in the final product that we get after different chemical reactions. Don't forget that we may make a product, but we may also have some intermediate reactions. Then it means intermediate products. So we may also need to quantify first identify and then to quantify those intermediate products as well. So all of these different activities are going to, again, allow us to write documents like the chemistry, manufacturing, and control, where we are going to give the proof to regulatory authorities that we are making the drug that we are talking about, that we can identify that we can also, again, purify that, meaning that we're going to be able to get or extract that drug from the mixture that has the drug or the substance in addition to different impurities. So we can also talk about the dissolution. So dissolution testing for a drug products or particle size determination for drug substance are also common. We can also talk about analytical method validation. That's a process of demonstrating an analytical procedure is suitable for its intended purpose. And of course, all of the information that we're talking about must be in the chemistry, manufacturing, and control document. Now, the purpose of the, the guidance is to provide recommendations on submitting analytical procedures, validation data, and samples to support documents to identify strength, quality, purity, and potency. So now let's continue with, American laws as well. Here we can talk about 21 CFR Part 312.50 and Part 312.94. So each NDA and ANDA must include the analytical procedures necessary to ensure the identity, strength, quality, purity, and potency of drug substance and drug product. So data must be available to establish that analytical procedures used in testing meet proper standards of accuracy and reliability. And here, of course, we already talked about pharmacopoeia. So the way, or these are the standards that are allowing us to test for the, identification and, of course, the purity of the products that we are making. So continuing with, the analytical procedures, the following list of information may be included in a description of an analytical procedure. Here we are always talking about the chemistry, manufacturing, and control document. It's a document that we're going to use in order to prepare regulatory documents that we're going to add into Module 3 of the CTD, format. And here, of course, we're going to look into the following information. So we are going to, have the principles of the analytical procedure, samplings, equipment. We talk about instrument types, detectors, column type, dimensions, and parameters like flow rate, temperature, and runtime. Of course, in next slides, we're going to go over different equipments as well, selected equipments, and we're going to learn more about those different instrument types, detectors, column types, etc. These are all information that we have to add into the chemistry, manufacturing, and control document, and then to use this in order to write regulatory documents that we're going to add later in different modules and sub-modules of Module 3 of our submission in CTD format. So we also have to talk about reagents, system suitability testings like blank titration, etc. We have to talk about the preparation of the standards, giving information on preparation of samples and procedures. Now, another important point is analytical method validation, which is the process of demonstrating that analytical procedures are suitable for their intended use. So data must be submitted to show analytical procedures meet standards of adequacy and reliability. So when we are talking about what an RA professional is doing, the regulatory affairs professional is going to receive the information from different sources within the company or outside by the suppliers, and then the person will need to understand, of course, different method validations or analytical procedures, because the regulatory affairs specialist is going to prepare the documents that we're going to add into the our submission in CTD format. So applicants based on, again, this guidance for industry, applicants should submit information on the validation characteristics of their analytical procedures, which may include specificity, linearity, accuracy, precision, range, quantitation limit or detection limit, etc. So they are all, of course, again, we can use, of course, ICHQ2, which is a key reference for recommendations on validation characteristics for analytical procedures. Now, let's go over a few definitions. Specificity, for example, is the ability to assess unequivocally the analyte in the presence of components which may be expected to be present, like impurities, etc. We talk about accuracy, which is the closeness of agreement between the value which is accepted either as a conventional true value or an accepted reference value and the value found. We talk about precision, which is the closeness of agreement between a series of measurements obtained from multiple sampling of the same homogeneous sample under the prescribed condition. And we can talk about detection method, which is the lowest amount of analyte in a sample which can be detected but not necessarily quantitated as an exact value. So now we have other definitions such as quantitation limit, which is the lowest amount of analyte in a sample. We have linearity, which is the ability to obtain test results which are directly proportional to the concentration. We have the range, an interval between the upper and lower concentration of analyte in the sample. We also have robustness, which is the capacity to remain unaffected by small but deliberate variations in method parameters. So now here we are again going over the information that we can find from ICHQ2R1. So in this table you can see several columns. The first one is the type of analytical procedure. So what again ICHQ2R1 is telling us is that we need to do the following validations in order to show, of course, that we are validating our procedures for making our drug. So we talk about different characteristics of the validation. We talk about the accuracy. We talk about the precision, about the repeatability, then about the specificity, about the detection limit by our equipments. We can talk about the quantitation limit, linearity, and range. So these are all different, again, characteristics that a regulatory affairs specialist will need to understand, because the information, again, is coming from different sources, as I already mentioned. And those information have to be understood by a regulatory affairs professional in order to use the information and add them into different documents that are used as regulatory documents for the preparation of our submissions in CTD format. And of course, those documents are usually used in Module 2, which is the summary of Module 3, 4, and 5, and in Module 3, which is related to the quality of the drug substance and drug product in the CTD format. Now we can now look into the ICHQ6A, which is related to the specifications and test procedures and acceptance criteria for new drug substance and new drug products. Here you see we talk about substance and products, but product is the substance plus excipients. So here we can again have the description about the again the drug substance. So that's a qualitative description of the dosage form that should be provided in size, shape, and color. So the acceptance criteria should include the final acceptance appearance. We talk about the product. So we talk about identification. So this is again this identification testing should establish the identity of the new drug substance in the new drug product and should be able to discriminate between compounds of closely related structure which are likely to be present. So again, I would like to repeat that we are talking about the substance, which is the active pharmaceutical ingredient, and we talk about the product, which is the substance, in addition to excipients that we are going to add in order to reach the final product. We can also talk about the assay, which is a stability indicating assay to determine strength that should be included for all products. In many cases, it's possible to employ the same procedure like HPLC for both assays of the new drug substance and quantitation of impurities. And impurities, these are organic and inorganic impurities. It can also be the results of degradation of the products and residual solvents that are included in this category. The residual solvents are solvents that are being used in order to purify our product. However, in the final collected, I would say liquid, for example, we may still have some residues of the solvents that we used in order to purify our product. Here we can also talk about impurities in addition to organic and inorganic impurities. And now, again, we can talk about the test for the drug products. The description is a qualitative description of the dosage should be provided. Again, the size, shape, and color. The acceptance criteria should include the final acceptance appearance. And the same thing applies as the one that we saw in the previous slide for the drug products, for drug substances, for the drug products. So identification, assays, and impurities. Let's talk about chromatography. This is one of the analytical methods in order to separate, again, different compounds in a final collected product, or it can be also an intermediate product. Here we are using different equipments. We're going to talk about the chromatography. First of all, with a simple one, and then talking about HPLC as well. We are going to see how those equipments are working in order to allow us to purify the active pharmaceutical ingredient or the substance, of course, after the production of a batch. So for the chromatography, I can talk about the fact that analytical techniques used for separating a mixture of chemical substances into its individual components are constituting the chromatography, so that the individual components can be analyzed. So we use chromatography to be able to separate individual components. Now, there are many types of chromatography. We have liquid chromatography, gas chromatography, ion exchange chromatography, and affinity chromatography. But overall, the chromatography itself consists in what we see on the right side of the slide. So we see that we have a column. We have, the element that we are going to add on the top of this column. The column is composed of a stationary phase that we are going to see in the next slides, what it is. It also has a mobile phase. So the element is going to be like, this is a mobile phase that is going to go through the stationary phase, and then we are going to see the separation of different components in the element. And at the end, of course, we are going to get the eluate, which is again the final product. And obviously, you may say, are we going to have one eluent and then one eluate? No. We are going to have, again, one eluent, but then the speed of the migration of different components in the eluent is not going to be the same. This means that when we are going to get or collect eluates, we may collect eluates at different times, and they are not the same compounds or components of that eluent. And this is why we use chromatography. So let's go over the next slides in order to explain this further. Now, first, let's talk about the key terms in chromatography, because when we are writing a chemistry manufacturing and control document, we also use these key terms, and they have to be included as well. So detector As the separated dye bands leave the colon, they pass into the detector. The detector detects each separated compound band against a background of mobile phase. So what's a chromatogram? It's a representation of the separation of several compounds that has chemically occurred in the HPLC system. A series of peaks rising from a baseline is drawn on a time axis. I'm going to show you, of course, several chromatograms as well at the end. Those chromatograms are going to give us the information related to each compound of our element at the beginning of the chromatography, but collected at different times. Then we talk about the mobile phase or carrier. This is a solvent moving through the colon, chromatography colon. We can talk about the stationary phase or adsorbent. This is a substance that stays fixed inside the colon. We talked about eluent, which is a fluid entering the colon. What is it exactly? We prepare our eluents. You may also have a chemical reaction or several even a series of chemical reactions, and at the end we are going to find the result of all of the chemical reactions, where in that fluid that we call eluent we have our API or active pharmaceutical ingredient in addition to impurities. So we are going to add the eluent into our chromatography column, and then all of the components that are in the eluent are going to migrate, or they are going to pass through the solid phase of the chromatic column. But because of their charge or their size or any other factors or variables that we can measure, we may see that those components, because of, for example, the size of their molecules, are not going to have the same speed for passing through the solid phase. Obviously, we are going to end up collecting several, eluates. These are fluid existing in the colon that is collected in flasks. We collect several eluates, and in each of those eluates, we are going to have, again, usually one component only. So it's the way to purify or to separate different components from an eluent. And then we also have analyte. It's a mixture whose individual components have to be separated and analyzed. So we use analytes. We are going to add it into the chromatography column, and then we are going to look into separating different components. Now here we can see the colon chromatography. On the left side you have again the loaded sample. So we now talk about the sample. The sample is what has to be analyzed, or I would say the sample has this a mixture of several components, including the drug or the substance that we made. Now we want to separate our substance from impurities because they are all in the loaded sample. Now we are going to put the loaded sample, and this is going to be on the top of the stationary phase in the chromatography column. Now, in the second picture from the left, we see that we are going to now add what we call a mobile phase. We put the mobile phase on the sample separation, on the loaded sample, and then we are going to see the separation, gradual separation, and we call that a sample separation. In the picture in the middle, we see that now we are having the mobile phase that is pushing several compounds in the loaded sample, and we will see that we have, for example, two types of compounds that have a stronger interaction with the stationary phase, and we have compounds that have weaker interactions with the stationary phase in the colon. Obviously, when we look into the compounds having a weaker interactions with the colon, we will collect them faster in comparison with the compounds having a stronger interaction that will take more time in order to pass through the stationary phase. This is one way to collect, of course, two compounds here. Now, we may also have several compounds, each compound having less or more stronger interaction with the stationary phase. And in this case, we will be able to collect different compounds having different level of interaction with the stationary phase. This is why when we look into a chromatogram, we will see that at different times we are going to collect different compounds based on their level of interaction with the stationary phase in the chromatography column. We can also use a high performance liquid chromatography or HPLC. Let's go first over the structure of an HPLC. First, we have the pump. So it forces a liquid, we call that a mobile phase, through the liquid chromatograph at a specific flow rate expressed in milliliters per minute. Normal flow rates in HPLC are in the 1 to 2 milliliters per minute range. Now, typical pumps can reach pressures in the range of 6,000 or 9,000 psi. So a pump can deliver constant mobile phase composition, we call that isocratic, or an increasing mobile phase composition, we call that gradient. Now let's go over what is an injector. The injector in HPLC introduces the liquid sample into the flow stream of the mobile phase. So typical sample volumes are from 5 to 20 microliters. The injector must be able to withstand high pressures of the liquid system. Now, the third important part of the HPLC is the colon itself. It's considered the heart of the chromatograph. The colon's stationary phase separates the sample components using physical and chemical parameters. So it's a way for us to purify our active pharmaceutical ingredient. Now, what's a detector? The detector can see individual molecules that come out, these are elutes, from the colon. Now, it measures the amount of these, so the chemist can quantitatively analyze the sample components, and it provides output to a computer that generates the chromatogram. Now, a computer controls apparatus and takes the signal from the detector to determine the time of elution, so retention time of the sample or qualitative analysis, and the amount of sample or quantitative analysis. Now let's talk about the HPLC packing materials. So what you can see on the right side is a representation of a colon. And in the middle you have the packing materials. This is again the stationary phase. And when we are going to again have our sample that will go pass through this packing material, we are going to see less or more interaction in between the material, in the packing material, and our sample. And what we see is that after the sample is passing through the package material, we are going to see several compounds that are going to get out of the HPLC at different times. So here we talk about the HPLC operation. So first, the sample is injected into the colon along with the mobile phase. So at the left side, you see the mobile phase at the bottom left, and then you see that it is injected into a solvent delivery pump. And then it goes through the injector that is now going to, again, it allows us the compounds to, again, achieve separation through varying moving speeds. So when you see the colon, of course, the first one on the left, you see that it goes through the detector, and then the next figures from left to right is showing you to what extent or at what speed we are going to see the separation of different compounds. So if you have, for example, an orange compound in the colon, we will see gradually that the orange compound becomes three compounds and then three, I would say, colors, because you have the compound that is represented here as a pink, and you have another compound represented as yellow. However, if pink and yellow are still mixed, then we see an orange color. This is why in the first column that you see, this is an orange mixture, then or sample. In the second column from the left to the right, we see the pink compound and the yellow compound, but in the middle of those, we still have that mixture. That's why we have the color orange. And gradually, when the cycle is rolling or going on, we will see that we are going to have two separated bands, one in pink, one in yellow. And here what happens is that when we are going to, again, get the signal, we are going to see the yellow band that comes first because it has less interaction with the stationary phase, and then we will see the pink band that is going to be detected. And of course, they can be collected as well. Okay, so now let's go over this using, of course, or adding to the system a computer as well. On the left side, we see that we have a solvent or a mobile phase reservoir. Now we inject it, and we are going to inject it to a pump. It's a solvent manager, and it's a solvent delivery system. Now we also have our sample. So while we are adding the solvent, we also add the sample that we are going to analyze, and that sample is also going to go through the injector or auto sampler, which is again a sample manager. So what is reaching the HPLC column? You're going to see your sample plus your solvent or the mobile phase that will both reach the first entry of the HPLC column that has the packing material. Now the sample with a solvent or both together are going to pass through the HPLC column from left on this image to the right, and then we are going to see that again, anything that is separated, those compounds are going to pass through a detector. The detector is going to send signals to a computer where we can see a chromatogram. We are having different peaks, and at the same time, it allows us, the detector allows us to collect different fractions. So those fractions that you see, number 1, number 2, and 3, in yellow, red, and blue colors, these are analyte fractions that are being collected. We also have, of course, in addition to different, again, analyte fractions, the waste. And the waste is, of course, here the solvent as well. However, don't forget that, and we talk about that too, is that in some preparations we may still have some waste or solvent as an impurity in our collected compounds. So in a nutshell, we understand now that we can use HPLC in order to purify our active pharmaceutical ingredient. All we need is just to know in this figure, for example, if we would like to collect number 1, number 2 analyte, or number 3. So what we do is that after a chemical reaction, we are going to have a mixture, a sample, a mixture of all of the compounds that need to be separated, because we don't want to use the collected fraction as is, as a drug. We would like to purify our API from that mixture or the sample. So we go over chemical reactions. We are going to get our samples. We are now going to inject the sample with this mobile phase into the HPLC column, and at the end we are going to find out which of the peaks that we can see on the computer corresponds to the active pharmaceutical ingredient that we would like to collect and then to package and then sell it on the market. So here you can see that your drug may be number 1, fraction 1, can be fraction 2, can be fraction 3. You also have some waste, but HPLC allows us to purify, to separate different compounds, and then to purify, again, our drug. And again, I repeat, your drug may be number 1, number 2, number 3, but these has been separated. They have been collected using HPLC. It has been separated, and now we have a purified compound. Now, for example, for the HPLC operation, again, here, this is, again, another way of illustrating, of course, this operation. This is a movie that you can watch on YouTube. I already gave you the URL that you can find at the bottom of this slide. Again, here it shows in a very simple way, again, how we are going to inject our sample and the mobile phase into our high pressure pump or HPLC from here for chromatography, and then we will see that that liquid or that fraction is going to pass through the high pressure pump, and then through the injector it's going to pass through the column, where we see here several compounds with different colors. They will all pass through the detector. Detector is going to tell us which compound is getting out of the column at what time, and at the same time this HPLC allows us to separate our active pharmaceutical ingredient or API from the remaining products or compounds in the same sample. Now this is an actual HPLC chromatogram. So you can see on the top left, for example, that we did have a sample, and the sample had several compounds. One of them is norepinephrine, we have norepinephrine, we have pseudoephedrine, we have doxylamine, we have pyrilyamine, we have dextrometorphan, and we have trimipramine. Now, if you would like, you have this in a mixture, which is in our sample. If you would like to separate them, we are going to inject our sample containing all of these compounds, and we are going to inject that into an HPLC. Underneath, obviously on the x-axis, you can see the time. So from the time that we inject, time 0, it will take 2.5 minutes before we get some impurities. Okay, but then at minute 7, we will only collect norphenylephrine, number 1. And then at the minute, I would say 6.5, we are going to get the norephedrine compound. And a few minutes later, we can collect pseudoephedrine. And it goes on, of course. And of course, after 15 minutes, whatever is getting outside of the HPLC will be trimipramine only. And this is, of course, one of the fractions. So from here, we know that as soon as we are injecting our sample in the HPLC machine, we will be able to collect several compounds at minute 6, 6.5, 7, 8.5, 11, 12, 50, and 15. And we know that each of these collected, I would say, fractions are corresponding to one of the chemicals that we have here. So from 1 to 7. So HPLC is allowing us to purify, to separate different components from one sample. And now imagine that at an industrial level, you are going to have tons of liquid where you have 5 compounds. Now you understand why in many companies, of course, big companies, you have 400 HPLCs working 24 hours a day. You're having technicians injecting the, I would say, those tons of liquids, for example, and they're going to use HPLC in order to extract or to purify the active pharmaceutical ingredient. And this is when we collect that specific compound. Of course, that compound is going to be the one, the product or the drug that we are going to use. That the compound we are going to, that fraction, we are going to isolate that, and then we are going to again do the necessary for preparation, maybe sterilization, and then for packaging, etc. Now here I talk about the size exclusion chromatography. So molecules, of course, must be freely enter and exit pores to be separated. Pores from the solid phase. So larger molecules elute first, followed by intermediate side molecules, and smaller molecules elute last. So here, if you have 3 types of molecules, A, B, and C, having 3 different diameters, we are going to expect that after having injected the sample that we have containing a mixture of A, B, and C, that we are going to be able to collect first C at time, for example, 10 minutes. Then we stop collecting. At time 10 minutes, we will be able to collect B. And then when we continue to collect at time 15 minutes, we will get the compound A. And again, this will depend here, for this case, on the size. Don't forget that we may have compounds that are going to be polar. In this case, we are going to use different HPLC columns. Some of them are based on the size, some of them are based on the, I would say, the charge of the molecules, etc. Now let's talk about the gas chromatography. The difference with HPLC is that here, instead of having a mobile phase, we have a gas. So we are going to have our sample, but what you can see at the bottom of the slide is that you have a carrier gas. This gas is, of course, connected to a gas flow or pressure regulator. And then you see now that we are injecting at the same time the gas as a mobile phase and the sample, a sample that contains several compounds, and one of them is the drug that we would like to purify or separate from that mixture. Now the gas and the sample are going to pass through a column, and then of course here we talk about the oven. It's another way to separate those compounds. They will pass through the detector. We will also collect the waste. We will also use a computer to look into the computer and data analysis, but at the same time it allows us to collect the product that which is the API from a mixture of those compounds in our sample. And again here, very similar to HPLC, here we can see again a gas chromatogram where, for example, if you have different names, like look, you have number 1, number 4, number 5, for example, you know that if your product, your API is number 5, the peak number 5 corresponds to the time when your API is getting outside of the gas chromatography. So it allows us, obviously, to again collect that compound at that specific time. The remaining, we collect them, but these are waste, so we just dispose them. What is infrared spectroscopy? Spectroscopy is the study of matter and its interaction with electromagnetic radiation. if infrared light is shined onto a molecule, the molecule can absorb energy from the light, and the energy from the light can cause a bond to stretch. This stretching is called a vibration. Now, the stretching vibration of a bond is like the oscillation of a spring, so a bond can be thought of as being a spring. So consider this bond, as you can see, and then imagine this bond in between those two molecules as a spring. Now, infrared spectroscopy is the most widely used experimental material characterization and analysis technique. So it doesn't allow us to separate things. Infrared spectroscopy is allowing us to, again, characterize a molecule. And what it does is that, in fact, because of the, the type of bond, we'll be able to look into several types of bonds. For example, a bond between a carbon and hydrogen, or a carbon and oxygen, or a bond between oxygen and hydrogen. So when we have the infrared spectroscopy, we will be able to characterize and analyze our compound. Now, how it works, transmittance of less than 100% indicates this specific frequency was absorbed by the molecule. And don't forget that each bond, a bond between molecules, has a different frequency of absorption. So that energy can cause a bond to stretch, resulting in a stretching vibration. Now, since signals in certain regions are diagnostic of certain functional groups, we can determine the presence of functional groups using IR spectra. This means that if you are going to analyze a product, an API, you can, by using infrared spectroscopy, you will be able to characterize the functional groups in that molecule. So if you have a big molecule like the one that we have here, you have a tunable laser. It's going to pass, of course, a light across the molecule. And of course, not just a molecule. This is I would say this is, you have billions of molecules, for example, in that sample. And then it will be now, absorbed by a detector. And the detector is going to show us signals, and those signals correspond with bonds in the molecule stretchings. It means that now we are going to be able to look into different functional groups that exist in the molecule that we are analyzing using infrared spectroscopy. Now here, for example, is an example of infrared spectroscopy. What you can see now is that, for example, on each of those, on left, I would say left upper spectrogram, you can see the absorbance on the y-axis on the left, and you can also look into the wave numbers. So what you can see is that if, for example, at the wave number 1800, we have CO, double bond, this is where we have much less absorbance. So if now for each and every of these functional groups we have the absorbance that is going to be lower, it shows that, for example, in this molecule we have 3 functional groups with CO. And then we have, for example, 6 functional groups with OH. We have 5 functional groups with CH. And we have 1 functional group with CO, like what you can see on the top right, spectrogram, or at the bottom, the other spectrogram. So what it does is that it's going to give us the number and the type of, again, functional groups, functional chemical groups. And then we will be able to analyze it. It means that we'll be able to see, okay, what is the formula of that compound, or to, again, test it or check it. What is a UV vis or VIS spectroscopy? So UV and visible absorption spectroscopy is the measurement of attenuation of a beam of light after it passes through a sample or after reflection from a sample surface. So what you can see on the right side is the sample, a few milliliters into a cuvette. And then we are going to put the cuvette into a device, a UV and visible spectroscopy. And then we are going to record the results. So we have in the device, we have the light source, we have an adjustable aperture, we also have the light that is going to go through the cuvette, so from the sample, through the sample. Then we are going to have an output, but this output is going to give us, of course, the absorption of that light. So UV spectroscopy is widely used for quantitative and qualitative study of analytes. It's useful for various types of chemicals. What we do is that we are going to add the analytes or the chemicals into the cuvette, and then we are going to look into the absorbance. Now, how we can use it? It's because if you have a reference, it can be you have a molecule that you made, but you have the reference of that molecule that is already made. It has been tested. We know that this is the valid reference. We will be able to use the reference, put that in a cuvette, and then put into a device, and then to read the output. And then we can use the molecule that we made. And we will again add that molecule into a cuvette, a new cuvette, and we are going to put that into the device. And then we are going to have, again, reading and output. If these are the same, it means that we made our product exactly as the reference that was made before. Now here, for example, this is an absorbance spectrum of an important biological molecule, which is nicotinamide adenine dinucleotide, or NAD+. Don't forget that each of those molecules, they have their own signature. They have their own absorbance spectrum. So what happens here is that, as you see, when we are going to look into the device, we are going to scan. So when you see here in this, again, a spectrogram, you see on the left side, on the y-axis, the absorbance that we see. And then we also see on the x-axis the wavelengths. So the device is going to pass, I would say yes, pass the different wavelengths in nanometer through the sample, and it's going to look into the absorbance of that sample for each, I would say for each and every wavelength. And then it's going to create this graph. So again, this graph is the signature of nicotinamide adenine dinucleotide. If you had another molecule, that would be different. What is microscopy? So microscopy involves microscopes to view objects and areas of objects that cannot be seen with the naked eye. So objects that are not within the resolution range of the normal eye. And of course, they are categorized, the microscopy is categorized into three fields: optical microscopy, electron microscopy, and scanning probe microscopy. Now let's go over the TEM, or transmission electron microscope. Although TEMS and light microscope operate on the same principles, there are key differences. In one we use the light, in another one we use electrons. In one we watch the light because we can look at that. For the electrons, we need to have a detector to detect the electron if it passes or not, and at what speed, and then we'll be able to create at the end images. Now, the main difference is that TEMS use electrons rather than light to magnify images. TEMS can magnify objects up to 2 million times. So the standard light microscope is limited by the wavelength of light and can magnify specimens up to only 2,000 times. Now here we can look into different TEM images where you see that they can, again, we can magnify up to 2 million times, and we'll be also able to analyze different structures as well. Now let's go over the scanning electron microscope, or SEM. The SEM also uses electrons instead of light to examine a much bigger variety of specimens. So the SEM has a large depth of field which allows more of specimen to be in focus at one time. So the SEM also has higher resolution, so closely spaced specimens can be magnified at higher levels. So on the right side of the slide, you can see the structure of a scanning electron microscope, or SEM. So you have on the top the electron gun, you have the electron beam that is coming from the gun, then here you have the anode, and then it will pass through a magnetic lens, and then we have the scanning coils where it will also go past through the again electron detector, and then it will then be I would say reflected to the specimen what we are looking at, and then from the specimen we are now going to collect the secondary electron, and we have the secondary electron detectors. Now the information from the scanning coil, from the electron detector, and from the secondary electron detector are all going to be analyzed in, of course, with a computer, and of course it's going to be again sent to a TV, to a scanner, and to a TV or a monitor. It's going to allow us to collect or get those images. Now here we can look into different examples of SEM images. Now, if you would like to compare TEM versus SEM, you see, of course, that again the resolution is different. SEM is much more; it gives us a much better, I would say, better picture, I would say, of the structure that we are looking at of the sample. What is a nuclear magnetic resonance, or NMR, equipment? This is a technique that exploits the magnetic properties of atomic nuclei. It determines the physical and chemical properties of atoms or the molecules in which they are contained. So NMR can provide detailed information about the structure, dynamics, reaction state, and chemical environment of molecules. So NMR spectroscopy is often used to investigate the properties of organic molecules. So again, as a regulatory affairs specialist, we need to know about these equipments, about these analytical tools and methods. That's because we may be dealing with organic molecules, inorganic molecules as well, and we need to be able to do the analysis to identify again those compounds, to separate those compounds, to look into their strength or the dose as well. And all of these different analytical methods are used by different I would say research and development labs, And this is the type of information that we receive in documents that we receive from different sources that are going to be end up in the chemistry and manufacturing and control document that we use in order to prepare regulatory documents to add into Module 2 and Module 3 for the, again, quality of the product that you're making. Now, this for the NMR again, it involves a thin glass tube used to contain samples. So typically tubes come in 5-meter diameters, but 10 meters and 3-meter samples are also used. It's important that the tubes are uniformly thick and well balanced to ensure they spin at a regular rate. So on the left side, you can now see an NMR equipment. Now, like again, other like chromatograms that we saw, for example, here we can talk about the examples of H1 NMR spectrum. So what is interesting here is that if you're looking into, again, a molecule of ethanol, that's what you see here with 2 atoms of carbon, 6 atoms of hydrogen and 1 atom of oxygen. We can see that we have in this molecule 3 different types of hydrogen atoms in terms of NMR. Now the hydrogen H on the OH group is not coupling with the other hydrogen atoms and appears as a singlet. That's what you see again in the middle. And then CH3 and CH2 hydrogens are coupling with each other, resulting in a triplet and quartet respectively. That's what you see on the left side and the right side. So for H, for example, you have a quartet, and on the right side you have a triplet. So by having again this type of analysis, we'll be able to look into the signal intensity versus chemical shift using the NMR. What are the enzyme-linked immunosorbent assay? Let's talk about ELISA, which is standing for enzyme-linked immunosorbent assay. It's a plate-based test designed for detecting and quantifying substances such as peptides, proteins, antibodies, and hormones. So in an ELISA, an antigen must be immobilized on a solid surface and then complexed with an antibody that is linked to an enzyme. So we can make those antigens against any type of peptide. So it is highly selective and again allows us again to do the detection. So detection is accomplished by assessing the conjugated enzyme activity via incubation with a substrate to produce a measurable product. So the key element of detection strategy is a highly specific antibody-antigen interaction. And again, we can make antibodies against any protein. And of course, we're going to use those antibodies in ELISA. Now the procedure for ELISA, here on the right side you can see it's first of all is coating. So polystyrene plate is treated with a solution of either antigen or antibody. Then we have the blocking phase. So an unrelated protein-based solution is used to cover all unbound sites on the plates. Then we have the detection phase, which is enzyme conjugated antibody or antigen binds specifically to the target antigen or antibody. And of course, if we, for example, add some fluorescence into that, I would say that environment, we can now read. So we remove the liquid and we wash the plate, and then we are going to read the results. So substrate is added and the signal produced by the enzyme substrate reaction is measured. So if, for example, we have a specific antibody that is going to bind specifically an antigen, here, like a vaccine, for example, we will be able to do the test and to, again, prove that, yes, we are having the molecules of our, I would say, yes, the components of our vaccine in our sample. But if the specific antibodies are not fixing any protein, it means that that are usually, again, they are targeted against the vaccine proteins, for example. It shows that no, the product that we have been making is not the vaccine that we are claiming to prepare. So the ELISA procedure is going to allow us, of course, to do the assays and to make sure that we are specifically having the molecules that, again, that we made. And again, this is also to ensure uniformity, of course, for those assays. Now we can talk about summary of steps in different ELISA types, because we have several types of ELISA. We have indirect, direct, sandwich, and competitive ELISA types. Now, for the sandwich, for example, we are doing the capture antibody coating. For the indirect, direct, and competitive ones, we have antigen coating. For, again, all of those, we can also have the blocking, etc., etc. Now, for the ELISA data interpretation, the ELISA assay yields 3 different types of data output. So don't forget, we are analyzing our products. Now, one of them is quantitative. So ELISA data can be interpreted in comparison to a standard curve, so a serial dilution of a known purified antigen, to precisely calculate the concentration of antigen in various samples. We can also have ELISA type of data output, which is qualitative. So ELISA can also be used to achieve a yes or no answer, indicating whether a particular antigen is present in a sample as compared to a blank well containing no antigen or an unrelated control antigen. It can also be semi-quantitative. So ELISA can be used to compare the relative levels of antigens in assay samples, since the intensity of signal will vary directly with antigen concentration. Now, this is again one way of doing the interpretation of the ELISA. So it can be, ELISA data can be graphed as optical density versus concentration. And overall, what we saw in this lecture was just to go over selected numbers of equipments that we use in order to, again, I would say, meet the requirements of the regulations. We look into 2 ICH guidelines, and we look into the fact that not only we have to have our analytical methods, but those analytical methods must be validated. And then when we are using any equipment, we are going to get the documentation for the again, for the methods themselves, for the equipments that we are using, as well as the validations that have been used. And we are going to, of course, provide all of these with all of these documents, the regulatory affairs specialist that is in charge of writing or using the CMC or chemistry and manufacturing and control document in order to prepare, regulatory documents for Module 2 and Module 3. Obviously, we didn't cover all of the types of equipments or all of the types of analytical methods. Again, I'm inviting you, of course, to discover more, to look into a different source of information. And I tell you that, again, this is not going to stop here. We are going to see more analytical methods that we are, or equipments that we are going to be able to use in, again, in the pharmaceutical field. Okay, so thank you so much again for your attendance, and I hope that you enjoyed today's lecture. Thank you.